Thank you, Gazette. Well, welcome to the first of our webinar series for 2021. This one is a little bit different from what we normally run. You know, you only need to log on to the internet or turn on the telly, and there's a new story about COVID-19 and the multitude of vaccines which have been approved or undergoing testing. It seems there are so many self-proclaimed experts with no qualification in this area who take great delight in telling you what their facts are, or heaven forbid, what their cure is, unsustained as they are. So where do we go to get the true facts? Well, we've gone to one of the most reputable, world-renowned organisations whom we regard as the source of truth in the world of vaccines and virus. Interprac has been most fortunate engaging with the Peter Doherty Institute for Infection and Immunity, who many recognise as a leading research organisation in the field of immunology and infectious disease control. Indeed, they were the first organisation in the world, well, maybe outside China, who managed to duplicate the virus. Even more fortunate, we've been able to engage Professor Terry Nolan to deliver this webinar to Interpracting friends. I won't go through Terry's extensive bio, but let me tell you, he's the Redmond Barry Distinguished Professor at the Peter Doherty Institute for Infection and Immunity. He leads the Vaccine and Immunisation Research Group. He's trained in medicine at the University of WA, paediatrics at the Royal Children's Hospital in Melbourne and Montreal's Children's Hospital, did a PhD in epidemiology and biostatistics. It just goes on and on his achievements and his qualifications. He's Honorary Senior Staff Specialist at the Department of General Medicine at the Royal Children's Hospital in Melbourne, Fellow of the Institute of Australian Academy of Health and Medical Sciences. He was the former chair of the Australian Technical Advisory Group on Immunisation, and he actually was engaged with WHO as one of the World Health Organization as scientific advisory group. Our presenter, Terry Nolan, is a big believer in education and is always, well, most of the time anyway, pleased to engage with people keen to know the facts, and that is why he's offered to deliver us this webinar. There's one thing that may be even more dangerous than a pandemic, it is ignorance. So what better way to dispel the myths, fictions, half-truths, and gain a true insight into COVID and the vaccine, but to introduce Professor Terry Nolan. Thank you, Professor.
Thanks very much, Mark, and hi everyone. Just doing a sound check first that, Mark, you can hear me, and also that you can still see my screen. Okay.
Yes,
just trying to get rid of this window here. On the screen, you'll see, other than the logos of the various organisations that I'm affiliated with, a picture of a fruit bat, who is the, notwithstanding what former presidents of the US have said, is the real culprit here, because the coronavirus originated in fruit bats. There's still debate about an intermediate host, whether it was a pangolin or something else, and certainly about whether it was actually in China or perhaps elsewhere in Southeast Asia that the virus emerged. But emerge it has, and as a result of that, we now, sorry, I'm just trying to advance my slides, Cosette, and nothing's happening here. Sorry, everyone, just a technical issue. Cosette, I've lost my mouse pointer, and also it won't let me advance the slide from the keyboard.
I can see your mouse moving.
I can't see it myself.
This worked perfectly 5 minutes ago, guys.
Yep, sorry, everyone. Let me just get this.
Isn't this just always the way? Let's see what we can do.
Okay, that's better. Whatever you've done has helped. Cosette, can you see the second slide?
Yep, it's all there.
All good now.
Okay, so just very briefly, everyone, what I'm going to say today are my own views and opinions. I don't speak on behalf of the Doherty, the University, the Murdoch, any federal or state government entity, any of the companies that I will be speaking about, or any non-government organisations. And I am a scientific advisor to a number of the major companies, including some of those that I'll be talking about with their COVID vaccines today. And I've also sat as a scientific independent member of what's called a data safety monitoring board for two of the major world companies that are producing vaccines, Serum Institute of India, which is the largest vaccine producer in the world, and Clover Biopharmaceuticals from China, and as well provide advice to other companies on vaccine-related matters. We at the Doherty have our own candidates. I'll speak briefly about them in partnership with Monash Institute of Pharmaceutical Sciences. And our funding comes from the federal government through the Medical Research Future Fund. I am chairing the Victorian Government's Advisory Committee on the rollout of the vaccine in this state. I have no equity interest in any vaccine company. Today's talk is in front of you, 10 broad headings, and it's going to be a bit of a roller coaster ride. And I just want to say, it's always very difficult to decide what level to pitch these sorts of things. And frankly, I've pitched it fairly high. I haven't dumbed it down. So my apologies if I do lose some of you in explaining some of the basic science issues and basic mechanisms. The slides will be available through Mark later, or at least most of them. And so there will be a chance, and some of the sources of cartoons and so on, which explain some of the biological processes, are available through some very good resources that are publicly available, in particular the New York Times website, which has superb animations and very good, clear explanations of processes and how things work. So we'll talk very briefly about the virus itself, briefly about the epidemiology and what's happening globally. Then we'll talk about the types of vaccines, the different platforms as they're called, how they work. The biggest part of the presentation will actually look at the results from clinical trials so far, with a focus particularly on the vaccines which the Australian government has secured for rollout here. The licensing and rollout now as it's happening around the world. How will the rollout happen in Australia? A very brief discussion of that, because those plans are still being formulated. The issue of evolving strains and what are called escape mutants is particularly topical and is going to become increasingly important in the future. So I will speak to that both in the beginning and then come back to it a bit later to explain what's happening with that. And also how we can tell if the vaccines, when they are introduced, whether or not they are affected by mutations and whether they're working properly. And then finally, an attempt, I know there'll be lots of questions, and these are the questions you can never answer. Journalists are always asking these. When will COVID normal transform into normal? When can we get back to some semblance of where we were before this dreadful global pandemic started? So firstly, the virus. Here is a cartoon of what the virus looks like. It's a single-stranded RNA virus. The RNA is the pink coils in the center of this sphere or envelope. And that carries the genetic information that allows it to replicate. It's not DNA, it's RNA. And on the surface of this is the crown, hence the name, the corona spike glycoproteins. They're basically a trimer, meaning 3 independent subunits coming together to form those spikes. And then in between some other elements in the membrane, but it's the spikes that we're going to be talking a fair bit about. Oh gosh, just lost again. I've lost my mouse and won't advance again. Cosette. Here we go. That might work. So here is a diagram, a 2-dimensional diagram. On the left, you can see that, if you like, a cut through of what I've just shown the cartoon of, and on the right, the fact that those 3 proteins are, if you like, bonded together to form this spike. But at the top, you'll see the binding site to the ACE2 receptor. This is the most important part of the protein, because this is what attaches the virus to the cell. I'll show you this in a minute, and allows the virus then to unlock the key into the cell, where it gains access to replicate itself. And that's when you get an infection. The virus fills the cell up and then is released from the cell and then goes and does it to other cells, and you get a massive infection. So most of the vaccines are directed against this protein. Some of the vaccines are really focused only on that receptor binding domain, RBD as it's called, but most are more broadly directed at the whole protein. Here is a linear, if you like, array. This as if we stretched that protein out and looked at all the amino acids that make up the protein. And the receptor binding domain is the green bit here. It's a very small part of the overall protein. I'll come back to that point shortly. Within that receptor binding domain, looking at this now, you'll see these letters here, this E484K, the N501Y, S477N. These are the mutations that have been occurring. And the ones we're most worried about are the ones that are occurring at the receptor binding domain, because that's where the virus attaches. If the antibodies that the vaccine induces don't prevent the virus from doing that because the mutations have changed the configuration, then the vaccine won't work or will be reduced in its effect. So that's why these particular mutations are very important. The mutants, there's now increasing evidence that these mutants, specifically the ones you will have read about in the papers, especially the one in South Africa at the moment and Brazil, that they actually carry 3 mutations in that receptor binding domain, the 417N, the 484K, and the 501Y. Those mutations decrease some antibodies' ability to recognize and neutralize the virus, as I've described. And there was a recent study, I mean, there are studies coming out literally every day at the moment, trying to understand what the significance of these variants is. But basically, one study looked at the whether that variant could be neutralized in the test tube by convalescent plasma, so people who had been infected with the original version of the virus and their blood had been collected at the time, so that they had antibody against the virus from a natural infection. When you introduce their antibody into a test tube, you can see whether it works against a mutated virus. And in fact, in about a half of the samples, their antibodies did not neutralize the virus. So this is one of the forms of evidence that gives us some concern, and I'll come back to this point later. Just a very quick cartoon. These cartoons are from the scientific journal called Nature, but basically on the left you can see here that, as I've described, this is the coronavirus with the spike protein, the RNA in the center, attaches to the ACE2 receptor. Basically, it's a lock and key combination on the surface of the human cell. The virus enters the cell, its RNA gets into the cell, it reproduces itself within the cell and then is released from the cell, and then basically is confronted by the immune system. So the immune system is 2 pathways, basically B cells, which make antibodies against the virus, and T cells and various other components of cellular immunity, including killer cells and macrophages, cells that actually destroy and eat up the cells, invading cells or viruses specifically, whereas antibody attempts to bind those viruses and neutralize them. So the entering the body, as I've just described, happens, and then the whole process, if you like, plays out in terms of the immune response. Now, let's just revert a minute to what's happening in the real world. So these are from the Financial Times. We have a very good website that gives accurate and daily updates with very good graphics on what's happening around the world, and you can pick individual countries around the world to see what's happening, similar to the Johns Hopkins website and New York Times, a number of others, but this is one of the clearest. So this is data only from September last year, so it doesn't show the first wave. But what it does show is the massive increase in cases. So these are actually deaths, and these on the y-axis of this graph, this is not a logarithmic scale. These are raw numbers rather than rates. And you can see here that the numbers of deaths per day that have been occurring has just gone stratospheric in the US. There's a slight suggestion it's starting to come down now. And similarly in the UK, very early suggestion that they may be over their hump. But compared to what has happened elsewhere in the world, I've put here Canada, China, and we're on the floor because we have had no deaths. Our total deaths over our entire experience is less than 1,000. So, you know, there are many thousands per day that are occurring in these countries. And this shows actual cases now, and these are rates of cases per 100,000. So to some extent, this adjusts for the size of the country, and the scale now is a logarithmic scale. So it's making everything look closer than it really is. So we are on the floor with China. China's done better than we have with a massive population, but we have done superbly. India, similarly, after having a horrendous start, has really started to bring things under control. Canada has not done that well, and the UK and the United States have basically been terrible. Let's now look at the vaccine platform. So broadly speaking, there are three types of antiviral vaccines. The first group are those which really relate to how to use genes, so either messenger RNA, mRNA, or DNA, and they encode the viral target proteins. So rather than having the protein itself, it's really the recipe for that protein, and then they use the host cell, the recipient of the vaccine, to make that protein, and then that protein is exposed to the immune system, and then antibodies against that protein are produced by the cell. There's some elegance about doing this, which I'll come back to shortly. The second are actually viruses. The most traditional and old-fashioned vaccine is a whole virus which has been inactivated, neutralized, if you like. These are less effective than these new platforms. The Chinese in particular have a number of these which are fairly advanced, and they're already rolling out. They're not bad, but it is a more traditional approach. A modification of that approach is what's called a vector, where you use a virus as a Trojan horse to actually introduce the viral genome, DNA in this case, into the human cell. And that genome has been re-engineered to allow it to express the target antigen, the protein of the virus, the spike protein. And then the cell basically does its work, as I mentioned before, in a similar way to what mRNA does. And then finally, protein-based vaccines, two groups. One is where you actually manufacture using a genetic engineering approach, the target viral protein, the spike protein, or some domain of it, such as the receptor binding domain, as I mentioned before. Or a modification of that is, in addition to having the protein, a process to help those proteins self-assemble into a virus-like particle. So they come together into a cluster and then make it look like the virus does, with a crown, if you like, of a coated surface of spike protein. And this can be done in vitro in modern systems, insect cells and other cells, to actually engineer those proteins, and can be produced at massive scale very, very quickly. So they're particularly important for a pandemic response. So here are the leading vaccines. There are many, many others, and I'll show you some numbers in a minute of all of them, but this is really the lead rank, and the history of how these were funded. So it's an interesting story, but there isn't enough time today to get through all of that. So Pfizer, BioNTech, the mRNA, and Moderna are the two mRNA vaccines you will have read about. Gamaleya is the Russian vaccine. That's an adenovirus vectored vaccine. The Oxford AstraZeneca vaccine, which you all know about, having read about in the press, is another adenovirus vector, although it's a chimpanzee adenovirus, whereas the others are human adenoviruses. CanSino, a Chinese candidate, another adenovirus. And the Johnson & Johnson, or Janssen, is their vaccine subsidiary, an adenovirus vaccine, which is one that may end up in Australia. Another Russian vaccine, which not as much is known about it, a protein. The Novavax vaccine, which I am going to talk about, because it is one that has been purchased by the Australian government. And then the Chinese candidates here, which are old-fashioned, but still nonetheless potentially valuable vaccines, but probably not as good as the others. So the ones which Australia has purchased are the Pfizer vaccine, the AstraZeneca vaccine, and the Novavax vaccine. Only one of these has been licensed so far in Australia, and that's the Pfizer. The AstraZeneca vaccine is expected to be licensed within the next couple of weeks. The Novavax is a few months behind, and they've only in the last few days released their preliminary phase 3 trial results, which I'm going to show you shortly. But I'm also going to show you a little bit of the Moderna and the Johnson & Johnson and Janssen vaccines, because the government has said it is talking to those companies, trying to procure additional supplies of those vaccines as further backup for us. And interestingly, the Russian vaccine, overnight, they've published in the Lancet, the premier medical journal, their results of an interim analysis of their phase 3. You remember that this was, Putin had, under an emergency use authorization, made it available to the Russian public several months ago. In fact, it was the first that was licensed in the world, but it had not even started a phase 3 study. They've now done a phase 3 study, and the results are looking very good, in fact. So how do these vaccines work, these different platforms? First of all, the nucleic acid vaccines. The RNA basically is synthesized. It's encased in a lipid coat, so a little globule of a refined lipid to allow it, when injected, to be released into the cell without being digested. And then it gets into the cell, and it doesn't go into the nucleus. A DNA vaccine on the left here, by contrast, does go into the nucleus, so that has its own complications, including that the administration of a DNA vaccine requires another process. An injection doesn't work, so you've got to use what's called electroporation. It requires a device which makes it much less suitable for mass use in a pandemic, and also much more expensive. But an mRNA vaccine is very simple, can just be injected. It gets into the cell, where it makes the proteins using the host's own cellular machinery, produces the spike protein. Then the immune system comes into play, responds to those spike proteins. These spike proteins are not harmful. They're components of the proteins, but sufficient for the immune system to recognize them and then produce both an antibody response and also a T-cell memory response as well. The second platform in the viral vector vaccines, I'm going to ignore the replicating viral vectors such as weakened measles, because the two candidates in that have dropped out in the last 2 weeks. They are from Merck. So the non-replicating viral vectors, the adenovirus, as I mentioned before, which is a common virus, either a chimpanzee or a human virus, has its own genes. They modify the genome to stop it replicating and also make sure it's not harmful, but include in its genome the code for the coronavirus spike protein. So the virus, when it's injected, is really acting as a Trojan horse, because it is able to gain access to the cell. The virus itself releases the genome into the cell, where, in the same way that an mRNA vaccine works, the cellular machinery then produces the target protein. So this on the left here is relates to a DNA, but not to an RNA. To this vaccine. Then there are protein-based vaccines, as I mentioned, simple proteins, and you'll notice that on the right here, the University of Queensland and CSL logo. That's the molecular clamp candidate, which used a subunit of the AIDS virus protein to clamp the three proteins together to keep it stable, which was necessary to get a decent immune response. And that development has been discontinued just before Christmas, because although the vaccine is looking very good, it lit up an AIDS blood test. And what it would have meant was that anyone who was vaccinated would have tested positive for HIV, which was never going to be something the public would accept. And so the government and the companies decided to discontinue it. A pity in a way, but they're working on whether they can have another approach without having that technicality. We at the Doherty are also working on a protein for the next generation vaccine. And then finally, the virus-like particles. So this is the Novavax vaccine. So not only is it a protein, but the proteins are designed so they self-assemble into this sphere with the proteins actually protruding from the outside. So it looks to the immune system like the virus and can make it an even more potent vaccine at stimulating the immune response. Okay, let's quickly now move into a description of these lead candidates. The first, this is a slide which explains what happened last year. The normal, at the top of this figure is the normal sequential phasing of doing clinical trials and all of the things that have to happen in parallel with that, actually sequentially in terms of scaling up for manufacturing. So the scientific bit of it is doing these clinical studies in humans. Phase 1 is the first trial in humans, really around safety. Phase 2 is really around getting the dose right. And then phase 3 is finding out whether the vaccine works and how well it works. What happened last year was, on top here, all these processes of manufacturing and so forth would wait until the phase 3 study had been completed to be sure they had a vaccine that worked. With significant injection of funds from the US government, the companies were asked to start manufacturing and gambling on the fact that their vaccine was going to work. So it compressed and brought all of those in parallel rather than in series. And that's how the shortening of all those processes was able to be achieved. There are other things that happened, but it's not cutting corners, but it is doing things which with much higher risk at the investment, the $1 billion of investment that is required to actually scale up all that manufacturing, not knowing whether the vaccine was going to be shown to be both safe and effective. This is from the New York Times website, which I encourage you all to look at from time to time. It's very helpful. This is just a top-line summary of 37 vaccines. This is pretty up to date in the last couple of days. 37 vaccines in phase 1, 24 in phase 2, 20 vaccines still in phase 3, 8 vaccines in some form of limited use with a conditional approval, 2 vaccines approved for full use only, and 4 have been abandoned along the way so far. So that gives you an idea. I won't go through on the right here what the specifics are of the individual phase 1, 2, and 3 studies and so forth. All of these clinical trials, the phase 3, this phased study, which is attempting to find out whether the vaccine works or not, are focused on a disease outcome. When you say it prevents COVID disease, we have to say, well, what sort of COVID disease? Does it prevent very mild disease? Does it prevent severe disease resulting in hospitalization? Does it prevent death, and so on? So the formality of a clinical trial is to codify all of this and pre-specify it before you do the study, written in a very technical, with a lot of detail in the protocol for the study, and approved by the regulator as well in the case of the in the US ones in particular. Most of the studies have taken this outcome as the principal outcome. So some of the vaccine efficacy numbers I'm going to show you in a minute are focused on any level of symptoms, so even in mild, but also PCR positive. So PCR is the genetic test to show the presence of the virus. So you have to have the virus, but you also have to have symptoms. Some of them have secondary outcomes or a co-primary outcome, which is more severe disease. And all of these medical words here really relate to severity, ICU admission, death, renal failure, and so forth. And also there are other outcomes which are secondary. In particular, this one here, this PCR positive for infection. This is important because we need the vaccines not just to prevent disease. We'd ideally want them to stop people passing the infection on from someone to someone else if they're confronted with the virus, because that's going to be very important in terms of being sure that if you've been immunized, you're not just protecting yourself, but you're also protecting from giving the virus to someone else if you've been confronted with it. And then finally, the safety issues are very important. I'm going to use the term vaccine efficacy. So without getting into too much of the maths here, the vaccine efficacy really is the difference in the new cases of the disease between the group receiving the placebo compared to the group receiving the experimental vaccine. So it's very simply 1 minus the risk in those who received the vaccine divided by the risk in those who received the placebo expressed as a percent. It boils down to, though, meaning that the vaccine efficacy of 95% means there is a 95% reduction in new cases of the disease in the vaccine group compared with the placebo group. So you need to take care when looking at these VE estimates, though, on what case definition they're using for the disease, because they're not the same. And generally speaking, we're going to see different VE estimates for severe outcomes compared to mild outcomes. And because the severe outcomes are less frequent, the clinical trials, even though they've been in many thousands of individuals, some of them still haven't got enough data to be absolutely confident about whether they're working, especially in the very elderly. So now this is what the scientific paper looks like. This is the first result I'm going to show you. This is the Pfizer vaccine BioNTech, which is the German company that had the technology that Pfizer partnered with. But basically, they showed here, this is the standard appearance here. Here we've got a column looking at different age groups. On the left is 18,000 subjects received the vaccine and 18,000 received the placebo. And here we've got the number of cases of, in this case, any COVID disease of any severity, and it had to be PCR positive for COVID, of course. So in this study, at the time in which this analysis was done, of the 36,000 subjects, there were 8 cases overall in the vaccine group of who met the definition of the case, whereas there were 162 in the placebo group. Same sample size, so the rates are comparable. That is, the denominators are comparable. But you can see here that this translates into a vaccine efficacy of 95%. This thing in brackets behind it is called a confidence interval. So that's an estimate of the precision of this estimate. So it's a very tight confidence interval. You know, it's unlikely to be less than 90%. The best estimate from the study is 95, but it may be as high as 98%. And then these are the numbers for the specific age levels within this study. So you can see here that we're particularly interested in the elderly here. They didn't have a lot of events in the elderly. There were only 5 cases in individuals aged 75 and over. It was still enough to show that not a single one of those cases occurred in the vaccine group, and all 5 occurred. That gives you a vaccine efficacy of 100%, but it's very imprecise because there weren't enough subjects really to be more confident about that. But you can see here in the slightly younger, older age group, 19 cases in the placebo group and only 1 case. But you can see the overall dominant profile here is that very few cases in any of the vaccine groups by age, and nearly all of the cases were occurring only in the placebo group. So pretty convincing evidence that this vaccine works very, very well. I'm not showing the safety data, by the way. In fact, today, just because of time, obviously the safety issue is of huge concern. The broad summary is that the safety analyses of all the studies so far have been very reassuring, and no major issues have arisen thus far. This is a map also from the New York Times, just showing where the vaccine has been licensed, either on the provisional basis, early limited or emergency use in orange, or green with full approval. And you can see that the Pfizer vaccine has EUA, emergency use authorization, in the US and Canada, Mexico, Argentina, Chile, and some others in South America. Australia, this is the only vaccine which is licensed here, and it's fully licensed in Australia. This does say early limited or emergency use, but the TGA have effectively given it a conditional full license. So it's a subtle distinction between an emergency use and what they've given it. Licensed in Europe and the UK and elsewhere, as you can see. The Moderna is the second of the mRNA vaccines, and its data, I'm going to go very quickly here because the data are very, very similar. The point estimate of vaccine effectiveness around about 95% is, again, and most importantly, and this is the case for all the vaccines so far, the vaccine efficacy against severe disease, so this efficacy of 95 is against any disease, but against severe disease is 100%. Not a single case of severe disease occurred in any of the vaccine recipients. And this is another 30,000 individual study. So that vaccine, is, we'll see in a minute, has also been licensed in the US. Here are the rates which look at the vaccine efficacy. This is another way of just looking at the blue square here is the point on the scale on the bottom of the vaccine efficacy. And we're looking at here by different groups. So for example, in particular, I'm focusing on age here, but you can see in the over 65s, they didn't have huge numbers either, but again, a pretty confident estimate of effectiveness, but pretty imprecise. And then similarly for males and females, very similar estimates. So nothing in terms of differences either by ethnicity. So Moderna's vaccine here, provisional license in Canada and the US, and also in the UK, and also in Europe and Switzerland, and also Israel. This is the AstraZeneca University of Oxford adenovirus vector vaccine. This is a chimpanzee adenovirus No time to explain why they've chosen a chimpanzee, but there is a strong reason why they did. These were their initial results, which showed confusing results because they had results from a British study and the Brazilian study, and the main US study still hasn't reported for this vaccine. They were late in getting started to recruit, so we're waiting on their major study for their final results. But these are the results that you've read about in the press, and in particular this issue that for various reasons, a sub-cohort of 2,700 individuals got a half of what the dose was they should have got for their first dose, 2.5 compared to 5. And the vaccine, ironically or paradoxically, the vaccine efficacy estimate was higher in those who got the lower first dose than it was in those who got the standard dose for the 2 doses. This is a 2-dose vaccine. So the Brazilian efficacy estimate was 62%, but the UK one was 90%. But it turned out that in this estimate, there were no elderly subjects at all. And there was a much different interval between the doses. And it turns out on closer analysis that increasing the interval between the first and second dose hugely benefits the immune response by the time of the second dose and also the vaccine efficacy. That's influenced the way in which the vaccine is now being rolled out in the UK. Interestingly, though, also in the British study, they were swabbing every single individual. Whatever it was, 12,000 subjects every week were getting a PCR swab because they were interested in seeing whether the vaccine prevented infection as well as whether it prevented disease. And this was relevant because, as I mentioned before, preventing infection is something that relates to herd immunity and the confidence that the vaccine is not just protecting you, but potentially will protect you from infecting others. And in the low dose group, the estimate of protection against infection was actually much better than people thought, nearly 60%. In the standard dose group, though, it wasn't nearly as good. So if this is the case when you use a larger dose 1, dose 2 interval with the standard doses, which the British are banking on, it looks like this vaccine will actually contribute fairly substantially to protection against infection as well as against disease. I'm going to skip this slide because it's too detailed, but basically it shows the same sort of numbers that I've just shown you. Now, overnight, the AstraZeneca Oxford people have released another paper also in Lancet, and this is a further data cut. So the same cohorts of both UK and Brazil from this vaccine, their new estimate across a pooled analysis across both the British and Brazilian subjects is the vaccine efficacy is 76% after only 1 dose. And you know that the British have decided they're going to roll out to as many people as possible. They're a very different situation to us because they have rampant disease. And they've decided they'll get as many people to have their first dose and worry about the second dose later. And so the efficacy after 1 dose is really important. This is actually really encouraging because it's looking better than the initial results looked. They still don't have a lot of severe disease in their study, which has been an issue with the German and announced today in the French regulators have said they're not prepared to approve the AstraZeneca vaccine yet because they haven't seen enough data in older subjects or with more severe infections. That doesn't mean they don't think it's going to work. They're just saying we haven't seen enough of the data. In the new analysis overnight, there were no additional hospitalizations or severe cases in any of the vaccinees, and there were 22 in the control group. So again, that's further evidence that this vaccine is working well. And as I mentioned before, this dose interval issue, vaccine efficacy after 2 standard doses rose from 55% with an interval of 6 weeks to 82% if that interval was 12 weeks. So it looks like the British will probably move to a 3-month interval between the first and the third dose, even though they've got rampant disease. They've done some mathematical modeling which shows that the risk of someone, because of imperfect or less perfect, if you like, vaccine efficacy, getting illness is offset by getting to a much larger proportion of the population. And then finally, the asymptomatic infection numbers also are further confirmed as being very encouraging. So this is where the AstraZeneca vaccine global situation is. Not licensed here yet, but as I mentioned, we're hoping that that will be the case in the next week or two. Licensed in Mexico, Brazil, Argentina, South Africa, India, Britain and the EU. And now Novavax, so this is the virus-like particle protein vaccine, which is a bit behind the others, and these results were released last week. So this is another small American biotech who have very, very good technology with other viruses that they had been working on, including Ebola and also RSV, respiratory syncytial virus. In their UK study, they had enrolled more than 15,000 subjects, so not as big as some of the US studies. They also have a US study which they're enrolling for at the moment, which will be in 40,000 subjects. Age in this study, aged between 18 and 84, and a quarter of them were over the age of 65. That's important to be sure that there are reasonable numbers in the older subjects. And the primary endpoint was the same as the others, so any disease with PCR confirming that it's COVID. And the first results based on 62 cases, of which 56 occurred in the placebo group and only 6 were observed in the vaccine group, means their vaccine efficacy estimate was 89%, also excellent. And of those 62 cases, 61 were mild or moderate, and only 1 was severe. So they need more events to be confident. This is just an interim analysis. They will have more severe events, which looks like they're going to occur in the placebo group as the study progresses. But interestingly, because they've been later with getting this study done than the others, these new mutant variant strains have appeared, and they were able to look in particular at what effect this had on the vaccine efficacy estimate if it was a mutant strain or the original strain, because they've got the genomic analysis on most of their cases. 56 of those 62, they knew which strain it was, and they were able to estimate VE specifically. And against the original COVID strain, the VE was 96%. And fortunately, against the UK variant, so this is what's called the B117 strain, it was still very, very high at 86%, but not as high. So this study was not designed formally statistically to test this, but this was indicative evidence that there might have been a subtle effect, but it may be no difference really, because that actual, in terms of these numbers, that difference isn't great. They also had a South African study with 4,500 subjects, which they produced interim analysis. They were interested in this study because HIV is prevalent in South Africa, although only 6% of the study group actually were HIV positive. Their estimate from this study of efficacy was 60%. Now, this is interesting because the South African variant during this study was widely circulating. And although the numbers aren't huge here, nearly all, 93% of all of their cases were this new escape variant from, or potential escape variant from South Africa that I mentioned before. So 1 severe case occurred, it was in the placebo group, and all other cases were mild or moderate. So they don't really have enough numbers yet. But first of all, 60% is still not bad. We regard anything over 50 as being a helpful and valuable vaccine. But clearly it's not the same as the 90% plus that was observed in the UK. Janssen, the Johnson & Johnson company, another adenovirus vector vaccine, which is different to all the others because this is a single dose vaccine. All the others are 2 doses. So if this was to work, there's going to be huge interest in this vaccine. And they announced 3 or 4 days ago their first interim analysis with a VE of 66%, but in this case, this is not against mild disease, this was against moderate and severe disease. So the VE against mild disease, which they haven't announced, would likely be less than this. They also observed, because they recruited subjects from US, Latin America, and South Africa, a slight difference in the VE estimates. This is another issue that relates to the circulating strain. Not a huge difference though between those estimates. And they, in terms of a more severe outcome, those severe and critical, the VE estimate was even better at 85%. There were no deaths in the COVID vaccine group, and there were 5 deaths that occurred. They were all in the placebo group. There were other deaths that occurred, but these are unrelated deaths. 16 in the placebo group and 3 in the vaccine group, but not related to COVID. And then these are the results of this morning. These results are literally less than a day old and published in the Lancet. So this is the Russian vaccine. This is also an adenovirus vector vaccine. It's a bit different to the others because they use 2 different types of virus, and adenovirus 26 for the priming dose and then adenovirus type 5 for the booster. But you'll see here the overall estimate. This is the same data layout that I showed you before. The vaccine group here in this column. Overall, 16 cases out of 14, nearly 15,000 subjects with, this is any disease, by the way, it's the same sort of outcome, versus 62 in the placebo group. And that gives the vaccine efficacy estimate of 91.6%. This is 1 dose, not 2 doses, 1 dose. This is excellent. And you can see by age, they didn't have huge numbers here over the age of 60, and they haven't given a breakdown of the over 75s, for example. But certainly in the over 60s, a 91.8% vaccine efficacy, so pretty uniform across the age ranges of the younger population, but really we don't know more about what's happening at the older age groups. And then finally on the bottom of this table, moderate or severe cases, there were 20 in total, of which none were in the vaccine group and all 20 were in the placebo group. So that's a vaccine efficacy against moderate or severe disease of 100%. They're great results. Interestingly, the Russian vaccine has already been approved, not just in Russia, but in Hungary, Algeria, Iran, Venezuela, Bolivia, Paraguay, Argentina. So this is an interesting vaccine. I'm showing it to you because there's a lot of rubbish in the press. Just because it's Russian, it can't be any good. The history of Putin approving it early under an emergency use, which as these results show, provided they hold up to scrutiny, of course, but as these results show, was not a bad decision to have made. It wouldn't necessarily have happened in other countries around the world. Licensing and rollout, what's happening around the world? This data is from Nature and actually shows the numbers that have been pre-ordered per head of population. So Canada had ordered enough for 9 doses per person in the eligible population, grossly in excess of what they'd need. They only need probably 2 doses per person. But of course, like all governments, they were hedging their bets and buying from a number, as we've done ourselves in Australia, from a number of providers in case things didn't work out. But you can see here that Australia was down here. This is prior to the UQ vaccine falling over, by the way. We were also very much in the high end of the distribution of purchase. And now the vaccine pre-orders by company. You can see here, broadly speaking, that AstraZeneca is really the dominant player. They can produce massive quantities. They have contracts with many companies, including the Serum Institute of India, to assist with production, also with us in Australia with CSL to assist with production. This is the Pfizer, Moderna, and the Russian vaccine here, and the others still in the process of being announced. Now, in terms of the rollout of the vaccine that's happening at the moment, these are the top 1, 2, 3, 4, 5. So these are numbers of doses administered per 100 residents. In Israel, the Israelis have really taken this well and truly by the horns, and I think something like 30% of their population are already vaccinated, received at least 1 dose with the Pfizer vaccine, I think. UAE, UK, Bahrain, and the US, the numbers of doses in the US, huge, but huge population. So 29.3 million doses administered. This is not distributed, this is actually in arms. So a massive effort in the UK, nearly 9 million, this is of a few days ago, has been massive. If you look at the rest of the distribution here, you'll see many other countries, and we're not one of them yet. We will be in a couple of weeks, I hope, but all these countries have started rollout of vaccines. So in total vaccination doses given globally, nearly 94 million. So that's the thing to keep in mind too with respect to the safety issue. These clinical trials were done with 30 or 40,000 subjects, many of them, which is a huge number, of which half would have received the vaccine. But now the numbers who are getting these vaccines with these tens and tens and soon hundreds of millions are going to give us much better data on whether rare events or other things that we might need to be concerned about are appearing. Finally, there isn't time to go into this fully today, but the COVAX initiative is an attempt to be sure that the developing world and countries that can't afford vaccines will have access to vaccines. So many companies in Australia have now subscribed to this initiative and are providing money, and the companies who've signed up are actually promising to provide vaccine into the COVAX pool to allow countries around the world to have access to the vaccine. How will the rollout happen here in Australia? So these are the ones, as I mentioned before, Pfizer, AstraZeneca, Novavax. In the right-hand side of this column, 10 million doses has been committed from Pfizer. That they are now in trouble with maintaining their volume around the world, not just to Australia, but to many other countries around the world. They've got some problems with the rate of production. AstraZeneca, 3.8 million doses made in Europe to be delivered this month here, and then 50 million doses to be made by CSL, and CSL's first vaccine is coming off the line, I think, next week, with a commitment to produce 50 million by the end of the year. Novavax, the one I mentioned, still a bit behind the others. 51 million doses has been pre-purchased by the Australian government. And it's been announced in the press, I have no inside knowledge on this, that the government is still trying to negotiate with both Janssen and also Moderna for further vaccine doses, just in case there are problems with supply of Pfizer or AstraZeneca or other issues with the clinical trial results, to give further insurance for our own supply. These are possible elements for a state-based COVID vaccine program. In Australia, we're going to see both the federal government and the state governments jointly sharing responsibility for this. The principles will be the same in most states, ensuring access for all, the vaccine being free of charge, enabling the highest quality and safety in the delivery of those vaccines, and also doing things to build and sustain public trust and confidence, making sure that communications from both state and Commonwealth are aligned. Basically, the distribution will be different because the Pfizer vaccine, I haven't gone into this in detail, requires freezing at -70 degrees or colder before it's distributed and thawed and then prepared for administration. The Pfizer vaccine will be preferentially used for the Category 1A, so I'm not showing you the around the country. There has been a very detailed process over the last 3 or 4 months to decide the sequence of individuals at highest risk and so on who will preferentially will be offered vaccine. So the hospital hubs will largely be responsible for the Pfizer vaccine, and then the other distribution for the AstraZeneca vaccine and Novavax ultimately through vaccine clinics, so health services, general practice, community health, local government, the Aboriginal controlled community health organisations. Also, at least in Victoria, thinking to have a more adaptive network to reach hard to reach communities that don't have good access to traditional services. This includes migrant and other communities, some who are marginalised, at risk and so forth. And potentially using a high volume pop-up image, which has widely been used in the US and also in the UK, to really get very large numbers through. The thinking about whether and how that'll be used is still happening. Rapid deployment, if there are outbreaks, to actually deliver vaccine when it's available. If there are localized outbreaks, to individual geographic regions. And we have a network of specialist immunisation clinics around the country to help with people who have complex issues with vaccine safety, or especially issues with hypersensitivity or anaphylaxis. I haven't gone into that today. That has emerged not as a major issue, but it is an issue for the mRNA vaccine in particular. Coming back then finally to this escape mutant issue, because it really is the issue of the day. These variants that I mentioned before, the UK variant of concern, the B117, has only one mutation in the receptor binding domain. The South African mutant has 3, the ones I showed you before. And the Australian mutant, we had the 501Y, had been, when we saw a lot more disease, was the one that was starting to appear here. Novavax, just as an example, with their results announced, they've already initiated development of new mutant strains with their recombinant protein. And they are really, if you like, anticipating that it will be necessary to roll out a modification of the target strain in their vaccine down the track. They argue, this is a quote from their head of research, Greg Glenn, that a primary benefit of their platform is that it uses very small amounts of protein. So they can do this fairly quickly and change. And every time they do this, they don't have to go back through phase 3 trials. The regulators will look at some minimal human data on antibody responses and so forth, and they will not have to go back over several months to these very expensive studies. How can we tell if the vaccines aren't working when they're rolled out? First of all, the test tube neutralization studies I mentioned before. There are data I've just shown you from clinical trials, but really the most important will be what are called vaccine failure analysis. Once these many millions of doses are rolled out, if the viruses are resistant to the vaccines, we will see vaccine failures. We'll see people being infected who've been vaccinated, and we will get a very good idea of, irrespective of what the test tube shows, that in the real world, if those vaccines aren't working. Finally, what does the future hold for a reversion to normal? The herd immunity issue I haven't mentioned in much detail. I referred to it in passing. That vaccine efficacy against transmission is important. In order to get herd immunity, we're going to need levels of coverage of at least 70% across the whole population. That's going to mean effective rollout of a vaccine that's also working. This could lead to the COVID passport idea that individuals who are effectively vaccinated may, subject to other requirements of testing and so forth, lead to increased mobility. This has happened with yellow fever around the world over many years, but it's not as a slam dunk because there is this issue of transmission. Are we going to see this virus mutate to this extent? We're all going to have an annual or every 2-year vaccination. It's looking like it's headed in that direction at the moment, but we don't know yet. One of the things that Peter Doherty has pointed out a lot is that the vaccine development has been so spectacularly successful. It sits alongside our spectacular failure to produce any antiviral drugs against coronavirus. If we had a single effective drug to protect against very severe disease, that would be a huge plus, and what we really need. The monoclonal antibodies, some of the others have not really, steroids, for example, are not that effective. We really need a very potent and effective antiviral drug. And then finally, these new platforms, what they've shown above all else is that this is the next wave for this technology for other viruses that, we've been struggling for years to get vaccines against RSV, respiratory syncytial virus, that causes bronchiolitis and may cause asthma, is an example. Cytomegalovirus and parainfluenza virus as well. Because they're so elegant and they work so well and appear now to be highly effective. And also vaccines against the flu, for example, it's likely the mRNA vaccines will be more effective than the flu vaccines we've got now. So just to finish, we've got 3 or 4 vaccines now with rollout to begin within weeks. There are still plenty of questions though. We don't yet know that you can reliably tell if someone's going to be protected if they have the vaccine, they have a certain antibody level. That's important for testing future vaccines. We're still finding out about how well these work in the very elderly, and particularly the frail elderly. We don't know whether these vaccines, irrespective of the mutation issue, will or will not need boosters if that antibody level, declines over time and needs boosting. The vaccine efficacy for severe and mild illness and for specific subgroups in differing transmission settings is things we still need to understand better. And as I mentioned, transmission implications for herd immunity. And in order to have proper rollout, we're still doing these very late studies in those who have immune deficiencies or are being treated with for cancer, for example, other things, adolescents and children, and also pregnant women. We haven't done phase 2 studies to be absolutely sure they're safe and what the right doses are in these particular groups. And then finally, this issue of the variant modified vaccines and whether that'll mean we'll mix the schedules up and have a priming dose with a conventional initial strain, and maybe the booster with the variant strain. That's it. Thank you very much. I'm sorry I've gone on a bit longer than I'd hoped.
Thanks, Terry.
Well, thank you, Terry. Yes, Cosette.
We just got a few questions, so we'll take a few of them. So the first question we have here is, are RNA-based vaccines safe with a high probability, specifically with regards to downstream impact upon the genome?
Yeah, so most definitely, and this is why they're very different to DNA vaccines. The RNA that's in this little globule of lipid that's introduced is degraded within a few days of its injection. It doesn't hang around. So what it does, it goes into doesn't even go into the nucleus of the cell. So it has no opportunity to interact with the genome at all in the human cell. And it's removed very quickly after it's done its job, which is to tell the cell to produce the spike protein and to let the immune system know, here it is, now you produce your antibody against it. That's how it works. So, you know, it's rapidly degraded, you know, in and of itself, very easy to administer. So both theoretically and now also in practice, from the evidence we've seen so far, it's looking like a very robust model.
Great. And then the next question we had here was, why are VRP vaccines difficult to manufacture?
The VLPs, the virus-like particles, because they require several phases. There are some at-scale technical issues around the different steps of producing the particles and then getting them to assemble. I can't give you the specifics on this, but it's not a massive difference, but it is not as simple and elegant as an mRNA vaccine is to change the script and then you know, put it into the system and have producing new vaccine within weeks.
Right. And then another question here that we have is, what is your opinion on the reason why Australia couldn't approve the Russian vaccine for use here?
I don't think it's been given a chance. I have no idea. I've heard that the TGA does have half a dozen vaccines in process of assessment for approval. I have no idea whether the Russian vaccine's been submitted. It might have been, but there would be, technically there's no reason why it shouldn't be suitable. The technology that's using is identical to the technology that's being used by the other adenovirus manufacturers. So if the Russians wanted to sell it to Australia, the first step would be for them to put it in front of the TGA and also have a discussion with the Australian government. Whether that's happened or not, I have no idea.
Got another question that's just come through, which is, are India and other non-Western countries fall in case because they're using HCQ?
Hydroxychloroquine. To be honest, I have no idea how prevalent the use of that is in India. We all now know on the basis of several studies that it's not helpful, doesn't really make a difference and shouldn't be used at all in the treatment. The reasons for out of control disease early in India were to do with rural populations, access, inability to effectively socially isolate, socially distance and so forth. The Indians have well and truly started to get their act together though, as the data showed that I showed before from the Financial Times graphs. And also India has, you know, India's one of these great paradoxes, a very sophisticated manufacturing sector and a huge wealthy or middle class, and then of course this massive poor proportion of the population. But in terms of their technology, the Serum Institute of India is a massive company. There was a fire in Pune last week at one of their new plants that they were building. Apparently that was not a plant that had anything to do with COVID vaccine manufacture. We're actually doing a study at the moment about to start in Melbourne with the Serum Institute of India vaccine with another University of Oxford candidate. I hadn't shown it to you today because it's further behind in its development. And we've done the phase 1 study with this. We're about to start the phase 2 study in Melbourne and probably Sydney as well, with what's called a recombinant protein. It's actually a receptor binding domain recombinant protein attached to hepatitis B surface antigen as a carrier. It's a very interesting prospect. But the Indian infrastructure for clinical trials and for all the rest of it, they're doing this in Australia because we don't have much circulating disease. We've formed a thing called Vax for COVID, which is an attempt to encourage the big international players to do some of their early phase studies in Australia, because you don't want to do those if there's a lot of circulating disease, because you don't want to contaminate the effect, if you like, of the vaccine producing antibody with the possibility of someone being infected in the general population. By contrast, the phase 3 studies can only be done in countries where there is a huge amount of disease, and the more disease, the better, because you get results much quicker. So that's why most of the phase 3s have been done in the US, in Brazil, in the Middle East, and other places.
Thank you. I think we probably have time for one more question. We've got one here which is asking, if you have, for example, the AstraZeneca vaccine now, can you then have a higher effectiveness vaccine like the Moderna vaccine at a later stage, or? Yeah,
that's a very astute question, because there are some theoretical reasons and also history with other vaccines for completely different diseases that having what's called a heterologous boost, a boost by a vaccine that comes from a different platform, may in fact give you a much better boost than if you use the same vaccine for the second dose. This was the case for Hib vaccines in the 1990s when they were first introduced, and there are immunologic reasons why that might be the case. The problem is having the data to be sure that that is the case. So people are going to be reluctant to start to do that without having some trial data. The University of Oxford have already announced they're partnering with Gamaleya Institute in Russia to do a combination a heterologous boost with using the Oxford vaccine and the Russian vaccine. Now, they are from the same platform. Obviously, it's even more interesting if the vaccines come from a different platform, as your questioner just suggested, an adenovirus vector as a first dose and then potentially an mRNA as the second dose. You won't find recommendations being made about those schedules until those studies have been done, though. And because the governments are going to control the distribution of vaccine, it won't be possible for people to do that themselves, in all likelihood.
Great, thanks for answering those questions.
Thanks, Cassette.
Thank you, Cassette. I'll tell you, Terry, after listening to your webinar, it fills me with a lot of confidence moving forward as we go through these turbulent times, this massive uncertainty with this pandemic and the virus, but to know that you and your team and people like you are working so hard and dedicated to making this world a safer place through your efforts. So I thank you for sharing your intellectual property. It's been really enlightening. I understood most of it. I'll have to get someone else to explain the business side of it. I'll ring you and have a beer with you later on. But it was a great webinar, and I think of immense value to us here at Interpack and our friends. So, Terry, please accept our gratitude and our thanks for a great presentation, a fantastic webinar. Thank you, Terry.
Thanks, Mark, and thanks very much for the opportunity. It's been a pleasure. Thank you.
Thank you, Terry.